Okay, so as Koei mentioned, I'll be talking a bit about how one can discover and evaluate scientific resources within dkNET, and one of the key things behind that are the research resource identifiers. So I'll give an intro there as well, and talk about some of the new things we're working on. So a product we're calling Resource Watch. And so why don't I jump right in? Koei already gave you a little bit of information about dkNET, but really, one of the things that dkNET was, meant to do was meant to be an information network, to connect, DK researchers and NIH-funded centers, and basically connect them to, resources and information that they could use. And so, Koei mentioned, a number of different areas here. I'll be talking about the top two, primarily the resource reports and the authentication reports. And so in the dkNET portal, those are found on the left side of the main portal area. That will allow you to jump right in and start using those tools. So a little bit about dkNET in the sense of how we support researchers through sort of the research life cycle. And so when one considers sort of research that you may be engaged in, you might begin by asking a question that requires some background research. There we can provide information through the discovery portal. You may then want to examine some data or construct a hypothesis for certain things. So if you're looking at certain types of genomic expression data, you might be able to go into the hypothesis center and work through some of your questions to develop or refine a hypothesis. Once you have a hypothesis, you will then want to plan your experiments. And here, this is really where you need to identify appropriate resources and also begin to prepare what's required by NIH for any grant submissions. So, the need to, specify, how you're going to authenticate, some of your resources or reagents. You will then, collect and analyze your data. Again, some of the tools in the Hypothesis Center for analysis might be useful, whereas, the resource reports are going to continue to be useful in terms of documenting via the research resource identifiers, what resources you're actually using. And then you'll be publishing your results. And with the new NIH data mandate, you'll also be publishing your data and sharing your data. And so this is where our fair data resources, will come in to help you and assist you with that. And so here's, a little summary of, what you might, do in terms of, hypothesis around, diabetes and eye problems. And so this is just a little example, that you can look at. So how do you describe reagents, right? So we all know, how you describe, recipes. You have, step one, which is the raw materials that you need, and then the methods, that are used to sort of prepare those materials. In practice, this is what, you see in, many older papers, where you have, some mice described, they're from Jackson Laboratories, but nothing else, is really there to help you identify what that is. And so if you go to Jackson Labs and try to search for this, you don't get any search results. So you can't actually figure out, what was being used. So how do you make sure that, when you have a published paper that which reagents and resources are used? And so, looking at in 2013 when we looked at that, it was hit or miss. If you could identify antibodies, cell lines, organisms, with the information that was included in papers at that time. And so this is where, we began to work on research resource identifiers, really to help identify, key biological resources. And so the RRID are unique identifiers. They're search friendly, and they are provided by independent community registries. So for example, Jackson Labs, their RRIDs are based off their IDs that they use in their catalogs. And the format for an RRID is very simple. The first part, RRID colon is, what type of identifier it is. The AB underscore means this comes from the antibody registry, and then the 1855196 is the identifier from the antibody registry. And so this way, we're able to federate identifiers from a number of different community authorities and use those then to unambiguously identify, for example, this antibody in the literature. And there can be other parts to the syntax. So again, I went through the RID portion of that, but that you may also want to provide the catalog number, for example, and the lot or version used. So again, there's in addition to the RID, you can also add some other defining information on top of that. So why were RRIDs created? One was for tracking. So how do we know what resources are being used and resources that are being produced by grant funding or by other centers, so that they could track what was being used. But a big part of this is also to help with rigor and transparency, right? So that there are these unique identifiers that can be tracked, where you can attach additional information about those resources that the RRIDs identify. And so nowadays in papers, what you'll see, for example, this is a paper from Nature. You'll see the antibody, the organisms identified by their RRIDs. And there are a number, these are now part of cell process star methods, and there are additional reporting guidelines that they are part of. And so these are sort of the publisher instructions from a number of different journals. And when you now go to databases and catalogs, you'll see that the RRID is provided. So the mutant mouse resource and research centers, you can copy it right from the portal. BioLegend, which is an antibody company, has those available, and also Cellosaurus, which is a community authority that is populated by the International Cell Line Authentication Committee, which actually goes out and aggregates information about cell lines. They also provide, for example, they provide the RRIDs as well. And then they are also, included in a number of RFAs. For example, NIDDA and NIDDK have a number of RFAs out that specifically require the use of RRIDs. So, how has this helped over time? When one looks at, when changes have been put into effect, there's an overall improvement in the literature. That's sort of the blue and yellow and red lines. But then there's, where journals such as eLife and Cell, so with STAR methods and other requirements, you see that there's an incredible jump in terms of, the ability to identify antibodies over, over the time from when we started the project. And that is due to, the reporting requirements, that are required. So, there are a number of ways that we then provide links to register the resource. So, for example, if you do have an antibody that that you're using, that may not be in the antibody registry yet, or something that is being developed, you can actually go to the antibody registry, for example, and register an antibody, and that will be curated and will be ready in a few business days for your use. Same thing with the registry. Some of the which is a registry of tools and software and databases. Cell lines, you would go through Cellosaurus and ICLAC, to register the cell line. For animals, you would, for organisms, you'd go to the specific community authority where that organism is managed. And then for NCBI biosamples, if you're registering biosamples, this is something you would do through NCBI, and there is some guidance we would give you there as well in terms of how that would need to be done. Okay, and the biosample example is actually one of the areas within dkNET where we actually have worked with the community to provide these identifiers, from, for example, pancreatic islets used by IIDP researchers. And so this is where we worked with, the IIDP and with NCBI, to develop a method, for NCBI to be the community authority for these biosample RRIDs. And, that is now part of dkNET. So again, we have a number of resources that we currently identify, but if there is something else, that you believe should be, identified through the use of an RRID, you can always talk with us, and we can, have a conversation about, if that is something appropriate for RRIDs, and then who, or what the community authority should be that provides the identifiers. So, the growth of RRIDs continues to grow, and it's, now part of a number of standards. So the, metadata standard that PubMed uses to import data, they're part of that, and the materials design analysis reporting checklist. So again, this is something that continues to grow, and there are a number of tools that we use to extract information. So this gets back to how do we get information about, usage of RRIDs, and also some, information potentially about, performance. And this is where we have developed an RRID robot called Cybot that goes in and finds information on RRIDs and papers. And then this is something that can be further curated and annotated. But the RRID provides the way for us to automatically and easily pick up the usage of these RRIDs in the literature. And again, once we have these RRIDs, as I just mentioned, that allows us to assemble this unique data set that aggregates information about these key research resources. So we have the sources and metadata about the resource itself. So this comes from community authorities. We then have information from literature. We can also tie that then to information from other projects. For example, the Human Protein Atlas has done validation on a number of different antibodies. I'll show you some other examples when I go through the demo. And then also knowing the literature, knowing authors, that have published using the RRIDs, we can also tie that into, other researchers locally, to where you are, that may have used that same antibody, for example, if you had some questions, right? So if you wanted to find someone local who you could talk to, about the use of the antibody. And we can also provide alerts, as we get information, for example, from Cellosaurus, that a cell line has been contaminated, that information, can be provided, through these, through different reports that we provide. And these resource ratings and alerts, for example, will show up on the resource reports, and we'll go through this a little bit through the demo as well. And they're highlighted, and then we also provide, for example, the Intestinal Stem Cell Consortium has done some validation or provided some information, for example, about this antibody. So resource reports. This is the primary jumping off point. And so they're a standardized report that provides information, about a specific resource, provides information on citations of usage and mentions, who is using them. As I mentioned, do they have issues? Is anyone familiar with them? What other resources? So on the right here, you'll see this blue graph, that shows, what other resources, are associated, with, this particular antibody. And then Resource Watch, which is, information on validation. So validation and performance information aggregated around RRIDs. And this will also allow for contributions from researchers themselves. So if a researcher, has done an experiment and it doesn't quite work out, because that may not get published and that information will then not appear, through Resource Watch, they could actually provide information saying, well, I tried this antibody, in this, in this specific preparation, and it didn't work as expected, and can, can provide information about that. So now what I'm going to do, which is always dangerous, is I will jump into demo. Let me go switch to Safari. So I have my different browsers here. So the resource reports. It's a little bigger. So we can go, and what we'll do is we'll start just looking at tools. To walk through a live example, I'm going to choose a well-known, well-used imaging tool called ImageJ. And so you'll notice that in the snippet, provides a little bit of information. It has information about, short description, the URL, but then also has, says that there are more than 10,000 mentions, of the use of ImageJ. And so if I click on, the resource itself, I'll be taken to the report. And, what you'll notice here, you'll notice a couple of things. So I'll scroll here. We have the main information on ImageJ here. You can easily copy the RRID, but there's also the ability to log in and claim ownership. And so ownership, if you have a software tool, you can claim ownership to that tool, and that allows you to provide updates to the description and the keywords, and you can also provide additional information about, if you have citations, which we don't have. We're only able to crawl the PubMed Central open access subset and other open access collections that we have been given access to. So again, it's not the full set of the open access literature that we have access to do text mining. And so, we can pull out information from what we have access to, but, owners can also contribute that information. You'll notice that on the right, there's a list of relationships. So within the registry that we maintain, there is a collection of other tools, for example, that ImageJ is related to, and then there's also plugins, for ImageJ, and so these are also all, the ones that we are know about are listed here, and so this is, directly from the relationships, that have been built up around ImageJ, the tool itself. When we look at all the information that's available, we also do try and track also funding if we have that information, like which institutes, which agencies have provided funding. And then there are also other potential IDs that have been used, for ImageJ that we provide. And so again, the citation and metrics I will link into now. And so if I go to the usage report, this is articles per year, and so you'll see that this is related to GraphPad Prism and a number of other tools like Fiji, Adobe Photoshop, which are all imaging applications, and MATLAB and R, which are sort of the statistical packages that you might be using. And so the mentions, if I look at the mentions, there is, we provide information. If it is something that is pulled out from a RRID specifically, this is something where we will say it's a verified RRID. And then we also have, other notations where, for example, through the name of the tool, or, for example, the URL. So we can tell the difference as to what the annotation was that derived the use of the RRID. And so again, this provides then information on the PubMed ID, and so you can actually then go back, directly from this list. You can look through the list and say, okay, is there anything that applies to my research? Another technique is, you can also use the RRID through tools such as Google Scholar, because the RRIDs are well formed and fairly unique. You can also do searches through the search engines as well, using these RRIDs. Now, since we do have the author information, about who is publishing using these. So, for example, if I were interested in image J, I could look for which of these papers have researchers that are in San Diego. So this is a way that I could further trim down the search results and try to find usage of the tool in my local community in case I wanted to touch base with somebody. So let me close that. Okay. Let me go back to the tools real quickly. Right. So if you do, we have the general search bar on top. You can search for whatever tools you like, and then just to give you a little orientation to the interface, we have both the snippet view and a table view. The table view allows you, to basically, do some additional. You can, sort. You can search within columns here. You can look, for example, at the different keywords that it's been tagged with. And you can also use these facets on the left. So, for example, if you're looking for, the resource type, and you're looking for, the software application, that will filter, this down based on the selection of that keyword. And, you can also look for resources where we have found mentions. So again, this is a way to filter something by potentially, being able to look at, what is, described in the literature, where you can, more easily read up on that. So that's just sort of a little orientation, to, the layout of these reports. And so what I'll do now is I will go back to the resource reports. And, we can go to cell lines next. So one thing you might want to do, and I want to show also sort of information on alerts. So if you have, a catalog number, from a vendor, you can type that directly in, and if we have that, if, for example, Cellosaurus has that catalog number ID, we will be able to direct you specifically to that catalog number. Now, what you'll notice is that there are multiple hits, and so there are other variants of this cell line, that either are under a different. So, for example, here is a different vendor identifier for the cell line. And so, when we do have information about a cell line or antibody being problematic, or the issues found, it gets applied, to all, versions, of that cell line. So now if I go into the the report, you'll see that that we have that information about that this is a contaminated cell line, and this is for the cell lines. This comes from the International Cell Line Authentication Committee. So if I were to search in cell lines, so one way to sort of go through and, for example, search. So let's say I'm looking for cell lines associated with the pancreas. So I can search, on pancreas, and so now we get a number of results. Now, some of them, what you might want to do is you want to first say, okay, well, I want cell lines that don't have any issues. So you can select as a facet that there are no known issues. If you further explore, you'll see that there's cancer cell lines, but you may not be interested in cancer cell lines. So you can actually refine the search. And here what we'll do is we'll add a minus cancer. So you can formulate queries similar to what you formulate in other search engines. Here I can go again to, okay, I want this with no known issues. And let's say I want to also find ones where I can read up a little bit about them in the literature. So I select that I want the mentions. And so now I have a set of resources of these cell lines, that have mentions in the literature. And so what I can do is I can, for example, let's go to this one. Just use the first one on the list. There are a number of defining citations that are given. There's a number of information. There are cross references to other databases, and you can then go and look at the actual usage of this cell line. And so you can look at those PubMed IDs and read up further along for that cell line. So that's sort of a common strategy that that can be used. There are also antibodies that have this type of usage information. So I can just do a very broad search. I can look at I get information. Then the first things I see are that there are a number of discontinued antibodies. I can use the same strategy that no known issues. But I can also look at ones, for example, where validation information is available. And so again, that's a facet, and that will provide links to antibodies that potentially have ratings or validation information. So if I go to that resource, for example, this one has validation information provided as part of the ENCODE project. Oops, let me go back. But let's say I want to then also have mentions. So I will tie those two together. And so, if I go into any of these resources, you'll see that here we have the mentions report available again. Oops, do that. And then we can, further explore, these different antibodies. This is where Intestinal Stem Cell Consortium has some information, and then there are the mentions in the open access literature. And again, if I go back to antibodies, again, there are a number of other facets. So if you're looking, to filter on the target antigen or the target organism, the clonality, the host organism, the vendor, you can all do that through the facets that are available on the left side. Okay, so that's sort of a brief overview of, how one can sort of, navigate, through the resource reports. We also have resource reports on organisms and plasmids and biosamples, as I mentioned. So these are 3 other types of reports, but we also, one of the newer ones that we've brought in are protocols. And so this is, there are a number of resources available now in terms of protocols that are available through sites such as protocols.io. So, for example, if one wanted to look at protocols, these are protocols from NIDDK sponsored mouse metabolic phenotyping centers, right? And so these are formally published protocols that are in protocols.io. So we have some basic information, and then we can link out directly to that DOI for the protocol. And this will take you to protocols.io and the published protocol. And so the nice thing in these published protocols is that they provide, a detailed, sequence of steps for the protocol. I'm not sure this one has any. Okay, so this also lists various materials and reagents, that are required. And so it's a really nice resource, for finding protocols. And this also allows you to copy a protocol. So let's say you found a protocol, that is for, the type of investigation you'd like to pursue. You can actually make a copy of that and then edit that copy based on, things that, you have had to change as part of the protocol. And then you can actually republish that protocol as sort of your, a version of that. And so again, these protocols, will also generate a digital object identifier so they can be cited in the literature. And so that's another unique resource that you can use, for your research. Okay, so that sort of covers, the resource reports. One tool that we have in beta is Resource Watch. And as I mentioned, Resource Watch is really meant to be a resource, for this performance information, validation information, issue information about resources. So for example, the search is, and this is, again, it's tied directly to the RRID. So again, these research resource identifiers are what is used, to bind all this information together. So if I type in, RRID for a cell line here, right, and then it will actually return, that information. So this is, the well-known HepG2 cancer cell line. And then, information that this is misclassified, that it was supposed to be a carcinoma, but was something else. And so again, specific information about this cell line. So again, it contains individual snippets, from various sources. And so again, we have one source of, basically 2 sources of information that we pulled information from that is basically the same in this case. So if I go to another cell line here, 1996. Okay, now one thing you'll notice here is, that along with the cell line, there are a number of vendors for this cell line. This has been noted as being contaminated. And this also provides, the PubMed references for that. And we also, for example, have information that, that certain vendors have discontinued, this cell line. So again, this is a tool to manage, these types of citations. Let me 78467. Let's say. I hope I typed that right. Okay, and now give a couple examples of validation information. So here's antibody. That's again, it's distributed by 2 vendors. So again, multiple vendors can distribute, basically the same antibody, and so they will receive the same RRID. And here you'll notice that we have two types of information. One is we have information from the Human Protein Atlas. Let's see while that loads. So, this is the information that they have, on tissues around that antibody. So again, you have to sort of look at that. So here we have the antibodies and validation, for that antibody. And then we have a new resource which we've been pulling in, which is Icaros or Icaros. And they are a group that is doing, and this resource does sort of large scale validation or performance information, generates performance information around antibodies. And so if you look at their reports, and so these are PDF reports, and so we'll link to the PDF report. These are published through Zenodo. But if I go into, let's see if I can make this larger. Yeah. So they'll have, the antibody characterization for, CD44. They'll actually go through and say, okay, well, these are the antibodies that were tested. And you'll see that there are a number of different manufacturers, the different RRIDs for the different, antibodies, from those manufacturers. And then, they'll, describe, the different figures and the different, screens that they've done. And so this provides a very rich resource for you to explore, antibodies or the characterization information provided by Icaros. Okay, let me go through one more example here. Oops, do AB 10013321. Okay, and so another area where we're pulling information from is from the literature itself. So now that we have, again, we've mentioned the critical nature of the RRID itself. So it allows us to bind information together, from various sources. Now, the other thing it allows us to do is to easily denote if a research paper, has used a specific resource. And we can then actually apply other text mining algorithms around that RRID. And so one thing, that we have done is, we have trained a natural language processing pipeline to extract information about non-specificity of antibodies, right? So we can pull out this information, and this is what we call antibody watch. So this was developed within our lab, and this allows us to pull out and provide this type of information. And so, there might be multiple statements, that are derived from the literature, but these are then stored in Resource Watch as something that is potentially useful to users. Okay, that covers sort of what I had planned in the demo. Let me go back to the slides here. Okay. And so I'll finish up by talking a little bit more about the authentication side of things. And so, NIH has published Rigor and reproducibility guidelines, and authentication of key biological reagents, resources is something that is required in new grant applications. And this is a one-page attachment that describes how you plan to authenticate these key biological and chemical resources, which include cell lines and antibodies. And so what is NIH trying to say is please pay more attention to your methods, right? So want to make sure that you're using good reagents and using them in the appropriate situations. And again, even though we know about the existence of contaminated cell lines, they're still used in the literature. Even though this is known, it is important that the researchers do authentication of their resources. And so for cell lines, ICLAC gives useful guidelines, and they also provide the list of these questionable cell lines. And so there are a number of steps that that ICLAC suggests, and we provide these through our authentication report within dkNET. Will provide you with these references and citations. And so, through the database, I did show you, where you can find information about things being problematic cell lines. So we went over that a little bit before. You can check out a paper published, from our lab that looks at, how has the use of RRIDs impacted, the use of cell lines, in the literature. Again, can't make any causal statements here because a lot of times, researchers will, use the RRID when, at publication time, but there is a decrease, in the use, of the RRIDs for problematic cell lines, when the RRIDs are used. But the goal is really, to get these RRIDs in the hands of researchers earlier, so that they can find out this information, so that the problematic cell lines, are not used in improper preparations, from the get-go. And then, same thing with antibodies. There are issues with antibodies, and there are different ways to authenticate antibodies. And so there was a publication in Nature Methods for proposal for validation of antibodies, providing a number of conceptual pillars, and all this is outlined in the authentication report section, of dkNET. But again, using the resource reports, you can look at, again, what we went through before. You can find the antibody. You can find people who have published on this. You can get information. If they've been discontinued. You can look for the validation information. So there are a number of things that you can do in prep work around the antibodies. And then the authentication reports are what tie this latest bit of information together. And so here you can actually add your resources to a report, and this will then provide any of the latest information we have. So if there are any alerts, they'll be displayed, and we'll also then provide this validation information and references to the source material about these various validation and authentication techniques. So that wraps up my whirlwind tour of dkNET's resources around resources, and I will take any questions.
Yeah, you can unmute yourself or use the chat to ask questions. Yeah. Mm-hmm. There is a question from the chat. Can I add my resource to the system? Or if I want to share validation data for antibody, can I add them to the system?
Yes, and so that at the very beginning of the talk, I did mention and walk through the ways that you would add different types of, for example, antibody information. And so for the antibody information, you would be taken to the antibody registry, and the antibody registry, you would enter your information there, and they curate their resources, and they would provide you with the RRID, and curators would get back in touch with you. For cell lines, for example, we would take you to the International Cell Line Authentication Committee through Cellosaurus. Yes, so there are ways that you can register those, but you would need to get in touch with the specific community authorities that manage that type of resource information. In terms of providing information about the performance of an antibody, for example, that's what I mentioned when I was talking about Resource Watch. So Resource Watch is the place where you could go and you could add your own information about a specific cell line or antibody.
Okay, thank you. There is another question. Let me see. I could not find a paper from our lab list under resource. Can I add a paper, or do I need an account?
That's a good question. I believe, can a user suggest?
I don't think in dkNET they can. I think they can in the other
Yeah,
interfaces.
I think we just allow it for the owners, but let me.
Yeah, but you can always put it in a comment at on your resource page. If you put it in the contact us, we will make sure it gets in.
I'm verifying that right now. Yeah. So I think, if you're an owner of a resource, you can right now. But if you are, so again, if it's a resource developed in your lab, then you can become sort of a caretaker for that record and provide those. Yeah.
Okay, and there is a question from Wenting. Very nice resource. I have a question on public data sharing. Can I get to know more information about data sharing and the download?
So data sharing. So you're talking about data sets that are available, because that
maybe it's being able to share that particular data, so download the contents.
Wenting, are you able to unmute? Are you able to?
Oh, yeah. Yeah, thank you for the talk. Yeah, my question is about for us to do the secondary analysis of the public data.
Okay.
Any chance to download those RNA sequencing or some in bioinformatics data?
Right. That would be associated more with the resources one could find through the discovery portal, where we link to various data sets.
Okay.
The other way is through the resource reports. You could find databases associated with various types of data, and then you could go to that database to get see what they have available for download.
Okay, I got it. Thank you. Thank you.
There is also another question from Sembra. How do we can you show us how do we access to Resource Watch? I'm not finding it on dkNET.
Yeah, so that is, again, that's it's been an internal beta, and we are releasing it soon. We will announce it through the main website. When we make that public facing, because we're doing sort of the internal testing right now.
So there's another question from the resource report under the mention. There is a verified and a verified RRID. Could you tell us what's the difference?
Oh, so the yes, this is in the mentions in the literature report. So the two were verified RD. I'd have to go back and check. What did you say, Koei? Verified RID and
verify. I think a verified RID is from the RID.
What was the? Oh,
yeah, I think maybe one of your slides including that.
Yeah, no, I did mention that quickly, but I want to make sure I'm using the right language for each one of those. So let me go to. And I can actually
And Jeff, you hasn't shared the screen. I just let you.
Yeah, no, I know. Let me. Oh my God, I need to go back to share my screen. Where's that? That's right there. Okay. Okay, so though there are two different options, right? There's a verified RRID, right? And you'll notice, if you can see my screen here where I've highlighted that blue. So this is a verified RRID, and you'll notice that there is an actual RRID in the snippet, so that comes out of the publication. And so here, this is something that has been brought out and been annotated by the SciBot pipeline. So this is a verified RRID reference, and so that is been verified. Where we see verified, this is where image J, for example, the name, or for example, the URL has been pulled out, and then this has been verified either by a curator or by the owner of the resource. And so that's what the verified means. And you'll notice that there are some that don't have either, right? So they haven't been curated in the sense that here image J is mentioned. But there it hasn't been verified, by a curator or a resource owner.